For today's audience, I think I don't need to explain how the interaction between trans-acting factors and cis-regulatory sequences control spatial temporal gene expression, because we know the gene regulatory elements such as enhancer, promoter, and insulators, they are often bound by many transcription factors and modified by different epigenetic modification. So this biochemical feature of chromatin has been successfully used to annotate and predict putative gene regulatory element in the genome. However, when I was doing my postdoc work with Bing a few years ago, I developed a method that we can do high throughput CRISPR screening to identify the functional enhancers from the in vivo chromatin context. I think one of the unexpected findings from the CRISPR enhancer screening is that we found about 10% of the gene regulatory element that are functional in regulating targeted gene expression, but we cannot find any known marks that are associated with the enhancer mark or gene regulatory sequence. So at that time, we named these enhancers as unmarked enhancer. And a similar observation was also made by another paper published around the same time, in which they also did a CRISPR screening. So when I started my lab, I became very interested in the question, what are the key proteins and regulators that determine the activity of gene regulatory element? About that time, around 2016 to 2018, several groups has published a paper that combines Cas9 targeting and a proteomics approach to do something like a reverse chip, so you can identify the proteins that are enriched on specific DNA sequences, and this method includes one which was developed by Eric Thornton's lab, which is also part of Folding Consortium. So when I started my lab at Duke in 2018, I recruited a postdoc, Tong Yu, trying to talk him into this idea to do proteomics analysis of enhancers. But I realized that as a junior faculty, sometimes it's a little bit challenging to convince postdoc fellow other lab members to take some high-risk project. Actually, when Tony asked me whether I know how to do proteomics analysis, I told him I had no experience of doing proteomics analysis. So we realized that, okay, doing this work to identify the protein bound to a single enhancer might be quite challenging. So how we can start this project? So we decided to focus on transposable element, which are the repetitive sequence in the genome. So compared to a single enhancer or promoters, actually transposable elements count for over half of the human genome sequence, and it's been known for decades that many of these transposable elements are the controlling element in the genome, and they exhibit spatial temporal activity. And also recent study by many labs, including several following labs, showing that the tight control of transposable elements activity is important for development and involving many human diseases. And most importantly, if we are able to establish this method using transposable elements, then will be technically will be 1,000 times easier than doing proteomics analysis on a single gene regulatory element. So to start this project, we focused on a type of transposable element called LTR7 HER-rich. So LTR7 HER-rich is a primary specific transposable element. There are more than 2,000 copies of LTR7 in the human genome. And a previous study has shown some very interesting features of LTR7 HER-rich. We know that LTR7 HER-rich, they are highly active in pluripotent stem cell, but when the human ESA start to differentiate, this element will be rapidly silenced. And also a few years ago, Bing's group showed that the LTR7 elements and HER-rich can serve as a tight boundary in the human genome. And also there are some studies showing that the LTR7 sequences can be bound by pluripotent factors and enriched for transcription co-activators such as the mediator and the P300. So we think that okay, LTR7 and HER-rich could be a very interesting element for us to begin our proteomics assay. So next, we are trying to find a way to efficiently target Cas9 to these LTR7 elements. To do so, we use a method called Causal that is developed a few years ago, that we can simultaneously express up to 15 guide RNAs from the single Causal construct. So we designed a 15 guide RNA to target the conserved sequence of all LTR7 sequences. And also we fused the dead Cas9 with a protein called a turbo ID. So turbo ID actually is a biotin ligase. It can biotinate the proteins that are spatially proximal to turbo ID bait. And the idea of cargo by ID is that we can generate the fusion protein of DCAS9 turbo ID and co-express DCAS9 turbo ID with a cargo construct that target LTR7 sequence. Sorry. In doing so, we're hoping to identify the proteins that are spatially proximal to DCAS9 turbo ID, which binds to the LTR7 DNA sequences. So Tongyu first validated the targeting efficiency of the cargo construct, and he did a DCAS9 chip-seq experiment, and we found that the cargo construct can efficiently target DCAS9 to over 73% of LTR7 sequences across the entire genome. And also Tongyu optimized the biotinylation condition, and we found that with 3 hours biotin treatment, we can efficiently label the proteins in human ES cell, but we can see a big difference between the cargo expressing cells versus the control cells. And we collect this biotinylated protein and did a mass spec analysis, and we are able to identify over 140 proteins that are specifically enriched in the cargo expressing cells. And when we look at the protein, actually, we are very excited because we found many proteins that are involved in chromatin organization, transcription regulation, epigenome regulation are enriched on LTR7 sequences. And initially, I asked Tongyu to focus on these transcription regulators, but Tongyu has his own opinion for his project, and he became very interested in a group of proteins that are known associated with the RNA methylation process. So actually, RNA is kind of similar to DNA. It can be also modified by chemical modifications. And one of the most abundant modification occurred on RNA is so-called m6A modification, and similar to chromatin modification. There are also m6A writers, erasers, and readers. And Tongyu became particularly interested in a protein called YTHDC2, which is RNA m6A reader. So he did a chip-seq experiment and found that the RNA m6A reader YTHDC2 is indeed enriched on the LTR7 sequences. And interestingly, the binding of YTHDC2 is highly specific for the active LTR7 sequences. And next, Tongyu knocked out YTHDC2 in human embryonic stem cells, and he picked 2 clones, and we found a very consistent phenotype, which is the deletion of YTHDC2 does not induce human ES cell pluripotency. I think this is important because I mentioned that the activity of LTR7 and HER-Rich is highly specific for human ES cell. So if knockout YTHDC2 that would have induced human ES cell differentiation, then it will be very challenging for us to study the regulation of DC2 on LTR7 HER-Rich. So the good news is that we can knock out DC2 without affecting pluripotency maintenance in the self-renewing culture medium. But interestingly, when we look at the expression of HER-Rich and HER-Rich related viral genes, we found that depletion of YTHDC2 leads to downregulation of HER-Rich transcripts. It has been shown that RNA modification is critical for the RNA stability for some of the transcripts. So we also measured the transcription rate, transcription of a newly synthesized HER-Rich RNA, and we found that the reduction of HER-Rich is mainly due to decreased transcriptional efficiency. We also did some experiment to show that depletion of YTHDC2 does not lead to HER-Rich degradation. So with this observation, we think, okay, maybe when we knock out YTHDC2, there are some transcriptional or epigenetic changes on LTR7 HER-Rich DNA sequences. And indeed, when we perform H3K9 trimethylation and H3K27 acetylation chip-seq data in both Y-type and YTHDC2 knockout human ES cell, we found that there is epigenetic silencing of HER-Rich LTR7 HER-Rich sequences, which is indicated by gain of H3K9 trimethylation and the loss of H3K27 acetylation. And next, Tongyu spent quite a lot of effort trying to figure out what's the biological significance of YTH-DC2 and LTR7 in regulating human ESL function. To make a long story short, he found that YTH-DC2 can prevent the neuronal fate of human ESL, because when we induce neuronal differentiation of both Y-type and YTH-DC2 knockout cell, you can see that the mutant cell express higher level of PAC6, which is a marker for neuronal fate. And similarly, we combine our cargo construct with the DCAS9 crab to induce global silencing of LTR7 sequences. And we found that the LTR7 silenced human ESL tend to express higher level of PAC6 when we induce neuronal differentiation. And the next, Tongyu was trying to identify the target genes that is responsible for the LTR7 and YTH-DC2 phenotype. So he analyzed the RNA-seq data that we generated from YTH-DC2 knockout, LTR7 CRISPR-I, and neuronal differentiation, neuronal cells that are differentiated from human ESL. And we found about 12 genes that are co-regulated by YTH-DC2 and LTR7, and these genes are also downregulated during the neuronal differentiation process. One of the transcripts that Tongyu identified is link-RR. So we tried to overexpress link-RR in YTH-DC2 knockout and LTR7 silenced cell, and we found that overexpression of link-RR can indeed rescue the neuronal differentiation phenotype of YTH-DC2 knockout and LTR7 epigenetic silencing. In the interest of time, I didn't show the data. Basically, we found a similar phenotype in the human ESL-derived embryonic bodies. So next, we try to understand why RNA M6 reader binds to the DNA sequence of LTR7. I think the most straightforward hypothesis is that the Hurwitz transcripts, which is expressed from LTR7 locus, are spatially proximal to LTR7 DNA. And if this Hurwitz RNA is modified by m6A, then there will be a chance that YTH-DC2 will be recruited to the methylated RNA. So will be YTH-DC2 will also be enriched on the LTR7 DNA sequences. So to test this hypothesis, we first examined the m6A RIP-seq data and YTH-DC2 RIP-seq data. And indeed, we found that the Hurwitz transcripts are heavily methylated by m6A, and also YTH-DC2 binding is highly enriched on the Hurwitz RNA sequences compared to control IgG antibody. As I mentioned, Hurwitz RNA is highly abundant in human ESL, so we want to make sure that the enriched binding of YTH-DC2 on Hurwitz is not simply due to the abundance of Hurwitz RNA. So we analyzed the Hurwitz sequences that are bound by YTH-DC2, and we are able to recover conserved m6A motif that are significantly enriched in the Hurwitz sequences that are bound by YTH-DC2. So next, to prove that YTH-DC2 can indeed bind to Hurwitz RNA, we synthesize 30 nucleotide RNA probe that contains the conserved Hurwitz sequence and also this m6A binding motif. And we synthesize two types of probe, one with m6A modification, one without m6A modification. So we incubate the probe, RNA probe, with the nuclear protein that we purified from human embryonic stem cell. And we found that the m6A modified RNA probe can efficiently pull down YTH-DC2, but not the probe without m6A modification. And to further validate the direct binding of YTH-DC2 and Hurwitz sequence that contains the m6A binding motif, we also purified the YTH domain of YTH-DC2 and performed the m6A experiment. And we showed that the YTH-DC2 domain of YTH-DC2 can specifically bind to m6A modified Hurwitz probe, but not the probe that has the same sequence but without m6A modification. And lastly, we ask whether there is a physical interaction between her-rich RNA and LTR7 DNA. So to address this question, we download the margin data that are generated by Zhongsheng's lab using human embryonic stem cell. And from margin data, we found that indeed the DNA sequences that are interacting with the her-rich RNA are highly enriched on the LTR7 sequences. And furthermore, we separate the LTR7 sequences based on whether these LTR7s are solo LTR7 or the LTR7s that are flanking the full-length her-rich coding sequence. And from this analysis, we found that the her-rich RNA are specifically intact with the LTR7 sequences flanking her-rich full-length sequence, but not those LTR7 sequences. So from this data, we demonstrated that indeed her-rich RNA are modified by m6A, and also there are physical interaction between YTGDC2 and her-rich RNA, as well as the her-rich RNA and LTR7 DNA. But to further test this hypothesis, we want to determine whether the m6 modification of her-rich plays a causal role to recruit YTGDC2 and regulate LTR7 epigenetic activity. To test this idea, we use a inhibitor called STM2457. So this is a m6A inhibitor can remove m6 modification from all the transcripts. So we treated a human embryonic stem cell using m6A inhibitor, and we found that after 2-day treatment, the m6A level of her-rich transcripts was reduced to like 20–30% compared to the control level. So we took this human ESL and performed YTHDC2, RIP-seq, ChIP-seq, and also H3 kinase trimethylation ChIP-seq data. So from these results, we can clearly see that upon global removal of m6A, there's a reduction of YTHDC2 binding on both RNA and DNA sequences, and also the removal of m6A transcripts will lead to epigenetic silencing of LTR7 sequences. So these results are encouraging, but we know that the m6 inhibitor can alter m6 modification of all the transcripts. So we want to make sure the observed phenotype is not due to some non-specific indirect effect. So to further test the function of m6A in regulating LTR7 activity, we utilize the targeted epitranscriptome editing technology. So basically, in this method, we can fuse the dead Cas-Rx with a protein called ALKBH5, which is the m6A eraser, and we can use GNRA to target this m6A eraser to the m6A peak of link-RR transcripts. As you can see from panel E, after targeted removal of after targeting this m6A eraser to the m6A peak of link-RR, we can effectively reduce the m6A modification level of link-RR transcripts. And after that, Tongyu did a ChIP-qPCR and a RIP-qPCR experiment, and he found that upon the removal of link-RR m6A modification, the binding of YTHDC2 on link-RR sequence was reduced, and also the binding of YTHDC2 on the 2 LTR7 sequences that flank link-RR coding sequence was also reduced. And important, without m6 modification of link-RR transcripts, we observed the gain of H3 kinase trimethylation of the 2 LTR7 sequences, indicating that m6A indeed plays a causal role to recruit YTHDC2 to LTR7 DNA sequences and prevent their epigenetic silencing. But from all this data, we still don't know why and how the m6A reader can regulate the chromatin activity of LTR7. So based on literature, I think one of the most effective mechanism to induce transposable element silencing is DNA methylation. And from our cargo by ID proteomics data, we found that actually TET1 is also enriched in LTR7 sequences. So we want to test whether YTGC2 can interact with TET1 and recruit TET1 to demethylate LTR7 DNA sequences. So Tongyu first confirmed that there's a protein-protein interaction between TET1 and YTGC2 by using endogenous protein co-IP experiment. And also we performed YTGC2 and TET1 chipseq experiment. And we found that both DC2 and TET1 co-bound on the LTR7 sequences. And we obtained this because there's no good antibody for TET1 chipseq. We obtained the TET1 human ESL line from Dan Wei's lab, in which Dan Wei knocking a V5 tag into the endogenous TET1 locus. And most importantly, when we compare the binding of TET1 in Y-type and YTGC2 knockout human ESL, we found that depletion of YTGC2 will lead to loss of TET1 binding on LTR7 sequences. And consistently in the YTGC2 knockout cell, we observed a gain of 5-MC modification and loss of 5-HMC modification on the LTR7 HER-rich sequences. And Tongyu also analyzed the data that are generated by Danwei Huangfu's lab, that Danwei did a RNA-seq and demethylation in Y-type and TET1 knockout human ESL. And we found that when Danwei knockout without TET1, the expression of level of HER-rich is decreased, and also the demethylation level of HER-rich got increased. Finally, to test the causal role of TET1 in regulating YTGC2 mediated LTR7 silencing, Tongyu did another very beautiful experiment. So he fused the dead Cas9 with the catalytic domain of TET1 and used cargo construct to retarget the TET1 catalytic domain to the LTR7 sequences in the YTGC2 knockout cells. And he found that the retargeting of TET1 catalytic domain can rescue the expression of HER-rich RNA. And consistent with this model, we also look at the changes of TET1 binding, 5-MC and 5-HMC using the epitranscriptome editing technology. And we found that when the m6A modification of linker R was removed, the binding of TET1 was lost on the two LTR7 sequences flanking linker R. And we also observed gain of 5-MC and loss of 5-HMC signal. So to summarize from all this study, we actually made all these findings based on a method that we recently developed called a cargo bar ID, that we can identify the proteins that are associated with transposable element sequences. And from these results, we show that YTGC2, which is RNA m6 reader, can regulate LTR7 epigenetic activity and human ESL fate. And mechanistically, we show this regulation is through the crosstalk between YTGC2 and TET1. After we submitted a paper, we thought we were the first one to who identified the crosstalk between RNA and DNA methylation. And also, I think the results are also quite interesting because we know the host genome has evolved so many different mechanisms to silencing transposable element. I think this is the findings that we show here suggest that actually TE can also utilize its own transcripts and also the host epigenome and epitranscriptome machinery to maintain their activity. And in the interest of time, I do not show all the data, but you can find more details about this work from bioRxiv that we deposited last year in September. And also, I'd like to mention that when our paper was reviewed, there's another paper published in Nature Genetics reporting the crosstalk between RNA m6A and DNA methylation. So I think the crosstalk between transcriptome, epitranscriptome and epigenome will be an interesting direction to further explore. And lastly, I want to thank all the team members who did all the work. And Tongyu really took a lot of initiative in this project. And over the past few years, to overcome many challenges that are caused by pandemic and set up all these new assays. And this project was also contributed by some lab members, including Yu Yuan and Jian Hong, who did some of the bioinformatic analysis. And we also want to thank our collaborators who help our BioID and many regions. And this work was mainly supported by the Genomic Innovator Award, which is the RCD5, and also some other grants that support our lab and research. So I'll stop here and take some questions.